Hi everyone. In this explainer, we'll be doing a detailed examination of a novel computational framework called Hierarchical Molecular Language Models, or HMLMs. This framework really introduces a new way of thinking about and modeling the complex, multi-scale information processing systems that are constantly at work inside our cells.
So the central challenge in computational systems biology has long been developing models that can accurately predict how cellular signaling networks will behave. Our established methods, like those based on ordinary differential equations or even Boolean logic, they run into significant limitations. ODE models, for instance, often require extensive parameterization. This makes them quite sensitive to initial conditions and often computationally intractable for large networks. On the other side, you have Boolean models, and while they are scalable, they tend to oversimplify molecular interactions, which means you lose critical dynamic and quantitative information. So it's become clear that a new approach is needed, one that can properly handle the context-dependent and hierarchical nature of these biological systems.
Our discussion today will proceed in 5 parts. First, we'll introduce the conceptual basis of treating cellular signaling as a kind of language. Then, we'll get into the mathematical formalization of this concept. Third, we'll describe the actual computational architecture of the HMLM. Fourth, we'll review its empirical performance on a validated signaling network model. And to finish, we'll discuss the future implications of this technology.
The foundational idea of the HMLM framework is to reconceptualize cellular communication entirely. It suggests that this intricate web of molecular interactions can be understood formally as a specialized language, one that has its own syntax and semantics that govern how biological information flows and gets interpreted.
This mapping provides a really direct analogy between natural language processing and molecular biology. So think about it like this. In a natural language model, the basic units, the tokens, are words. In an HMLM, the tokens are molecules, things like receptors and kinases. The syntax, which is the set of rules for combining tokens, corresponds to the biophysical principles of how molecules interact. Semantics, which is what gives everything meaning, is represented by the functional outcomes of those interactions, like protein activation or inhibition. And finally, discourse, which is the formation of a whole narrative, is analogous to the coordinated activity of a pathway that leads to a specific cellular response.
Let's move now from that high-level analogy to the mathematical principles that give the model its rigorous foundation. This formalization is really the critical step. It's what allows us to apply powerful computational techniques for machine learning and information theory directly to biological systems.
The basic building block of this whole framework is something called an information transducer. You can think of this as a mathematical abstraction for any biological entity, whether that's a single protein, a large complex, or even an entire organelle. It's formally defined by a tuple with 5 components: an input space, X for incoming signals, an output space, Y for outgoing signals, a multidimensional internal state space, which is S, a state transition function, F, and an output function, G.
Now, that internal state space, S, is a crucial component. It's designed to capture the complete condition of a biological entity at any given moment. This is a high-dimensional space that includes things like conformational states, so the protein's 3D structure, its activation states, its binding states with other molecules, any post-translational modifications like phosphorylation, and even its location within the cell. This comprehensive representation is really key to the model's ability to make accurate predictions.
The dynamics of an information transducer over time are governed by 2 core equations. The state transition function, f, tells us that the entity's state at the next time point, s of t plus 1, is the function of the current input, x of t, and its own current state, s of t. The output function, g, then determines that the transducer's output at time t, which is y of t, is the function only of its current internal state, s of t.
So, these individual transducers are then assembled into larger systems. An information transduction network, or ITN, is defined as a directed graph where each vertex is one of those information transducers. The directed edges between them represent the information channels that link the output of one transducer to the input of another. This is how you build a topological model of a complete signaling cascade.
One of the key innovations of the HMLM framework is how it explicitly handles biological scale. It does this using three mathematical operators. The aggregation operator combines information from a lower level to a higher one, for example, from the states of individual proteins up to the activity of an entire pathway. The decomposition operator does the reverse, and the translation operator is used to convert information between different formats but at the same hierarchical level.
All right, so now we'll look at how this theoretical framework is actually realized in a computational architecture. The model is based on the transformer, which is a deep learning architecture that has really set the standard in natural language processing. Here, though, it's been specifically adapted for the graph-structured and multi-scale nature of biological networks.
The computational process itself involves four main stages. First, a multi-scale embedding function takes the discrete nodes of the biological network and maps them into a continuous vector space. This encodes information about the entity's type, its features, and its position in the network. Second, a graph-structured attention mechanism calculates the relative influence that connected nodes have on one another. Third, temporal integration introduces a time-dependent component to model the system's dynamics. And finally, a prediction layer uses all of this processed information to output quantitative predictions.
At the very heart of the transformer is the scaled dot-product attention mechanism. Now, in this biological context, the query, key, and value vectors, that's Q, K, and V, are projections that are derived from the node embeddings. The query vector represents the current node state. The key vectors represent the states of its neighbors. The dot product between Q and K determines the relevance, or what we call the attention score, between the nodes. These scores then act as weights for the value vectors, which contain the neighbor's information content, to produce an updated, more informed representation for the original query node.
So how well does it work? The HMLM framework was empirically validated using a comprehensive, manually curated signaling network model of a cardiac fibroblast. This cell type is integral to cardiac tissue homeostasis and fibrosis, making it a relevant test case. This next section will present the results of the model's predictive performance compared to other established methods.
This chart summarizes the primary performance comparison. We're looking at temporal signaling predictions across 16 time points. The metric here is mean squared error, where a lower value means higher predictive accuracy. As you can see, the HMLM achieved an MSE of 0.058. This is a superior result compared to both a standard graph neural network, which had an MSE of 0.083, and a calibrated ordinary differential equation model, which came in at 0.121.
To put that into perspective, the lower mean squared error of the HMLM corresponds to a 52% improvement in predictive accuracy when you compare it directly to the benchmark ODE modeling approach for this specific task.
A particularly interesting advantage of the HMLM architecture is its performance when data is sparse, which is a very common scenario in biological experiments. When it was trained with only 4 time points instead of the full 16, the HMLM maintained a low MSE of just 0.041. In contrast, the performance of the other models degraded significantly, which indicates the HMLM is more robust and data efficient.
Beyond just predictive accuracy, the model's attention mechanism gives us a degree of interpretability. It highlights the interactions that were most important for its predictions. For instance, the model assigned high attention scores to known canonical interactions, like the TGF-beta receptor activating SMAD3. But it also identified and quantified the importance of cross-pathway communications, like the link between SMAD3 and YAP, which is a known point of integration for signals in fibrosis.
So, having established the conceptual framework, the architecture, and the empirical performance of HMLMs, we can conclude by discussing the broader implications of this approach for the future of biological research and potentially for precision medicine.
The potential applications here are quite numerous. This technology could be used to generate patient-specific signaling models from clinical data, which would allow us to predict individual therapeutic responses. For complex diseases, it could help identify optimal multi-target intervention strategies. The models could also predict mechanisms of drug resistance by simulating how networks adapt over time. And more broadly, it could accelerate the cycle of scientific discovery through rapid, in silico hypothesis testing.
Of course, it's also important to acknowledge the current limitations. The framework's reliance on pre-existing, curated network topologies could introduce a confirmation bias, potentially constraining discovery to only known pathways. So, a primary area for future work is developing methods that can infer network structure de novo, directly from data. Other critical directions include integrating different multiomics data types and perhaps developing large-scale pre-trained foundation models for cellular biology.
The effectiveness of this language-based approach to modeling cellular signaling brings us to a final, more speculative question. It raises the possibility that universal, language-like principles might govern information processing not just in our cells, but across many scales of biological organization. It suggests there could be a fundamental correspondence between the structures of information in language and in life itself.